analysis of proteins and macromolecular complexes. I'm Dr. Chad Schwartz, Senior Application Scientist at Beckman Coulter, and I'll be your moderator for today's event. We are delighted to bring you this educational webinar presented by LabRoots and sponsored by Beckman Coulter. Beckman Coulter is a leader in centrifugation and flow cytometry and has long been an innovator in particle characterization, lab automation, and genomics. Beckman Coulter products are used at the forefront of important areas of investigation and discovery. For more information, please visit beckmancoulter.com. A few announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the icon in the lower right-hand corner of the slide window. If you have any technical problems viewing this presentation, please click on the support button at the top right of your presentation window or submit your problems to the green Q&A button. I am pleased to introduce today's speaker, Dr. Andrew Herr. Andrew Herr, PhD, is an associate professor in the divisions of immunobiology and infectious diseases and the Center for Systems Immunology at Cincinnati Children's Hospital Medical Center. Before his recent move to Cincinnati Children's, he was an Ohio eminent scholar in structural biology at the University of Cincinnati College of Medicine. Dr. Herr trained in molecular biophysics and structural biology at Washington University in St. Louis and Caltech before joining the faculty at the University of Cincinnati. Dr. Herr's laboratory uses a combination of analytical ultracentrifugation, X-ray crystallography, and a wide array of biophysical techniques to analyze the structure and function of proteins involved in bacterial pathogenesis and the host immune response. For example, they have been studying protein assembly events and underlying intercellular adhesion in bacterial biofilms. In this project, AUC data provided the critical clue leading to a novel antimicrobial technology under development for therapeutic use. The Herr lab has used AUC to study proteins that range in size from small polypeptides to massive antibody immune complexes. Integral membrane proteins, intrinsically disordered proteins, and complicated assembling systems. They have also worked with biotech and pharma companies to analyze protein samples and help guide formulation decisions. Dr. Herr has authored over 50 published manuscripts and book chapters, with over half of these involving AUC analyses. I will now turn it over to Dr. Herr for his presentation.
Thank you, Chad. A very kind introduction. I'm very pleased to be able to do this today. I want to talk today about the use of analytical ultracentrifugation, which is an incredibly powerful and useful technique, and highlight some ways that it can be of use to structural biologists in particular, but also more broadly to other people doing protein chemistry, biochemistry, pharmaceuticals, and biotech. So, what is analytical ultracentrifugation? Well, it's a technique that was invented in the early 20s by Thijs Svedberg, who later went on to win the Nobel Prize. I would argue, and many here would probably agree, that it is the best solution-based technique for looking at protein mass, assembly state, and getting equilibrium constants for oligomerization processes. It's also very useful for characterizing solutions, characterizing samples, looking for polydispersity or aggregation, as well as things like conformational change, formation of protein-protein complexes, and even highly complicated mixtures, which we'll get to toward the end of the seminar. There are two primary approaches one can use in the analytical ultracentrifuge. We'll be talking a lot about sedimentation velocity, and then we'll also talk part of the time about sedimentation equilibrium. So here is the basic cutaway view of what the inside of an analytical ultracentrifuge looks like over here on the right. I'm going to turn on my whiteboard here so I can make some notes. What you'll notice is on the right-hand side, right over here, is an illustration of the rotor. This is what contains the samples. This diagonal tube here is a monochromator, and the light passes up from a xenon flash lamp underneath the chamber, and then it shines down through the sample cell, and you can actually measure the absorbance or the interference pattern of a sample as it is spinning at up to nearly 60,000 RPM. So it's all done in real time. And this shows you, pardon me, that there is the interference optical system starting here and going down. And so it's a very complicated arrangement, and even though the ultracentrifuge looks essentially like a washing machine or a standard ultracentrifuge, there's a lot going on under the hood. On the lower left here we see this is an example of an XLI. This is the Beckman higher end analytical ultracentrifuge with both absorbance and interference optics. And up here, this is what it looks like when you look into the sample chamber. There's the rotor in the center, and what we're looking sort of past here is the monochromator, the ascending limb, and then over here, this is the laser that will be used for interference optics. Here's another nice view. Many of these figures, by the way, were provided by Beckman, so I appreciate that. This shows you the schematic for absorbance optics of how you have the light coming from the xenon flash lamp down here. It bounces off a toroidal diffraction grating which selects the wavelength, and then it shines down through the sample. And so the light that passes through the sample is then passed through this radial scanning setup. So basically there's what's called a slit assembly. There's a narrow slit that lets only a small bit of light through, and this lens in the slit assembly will move back and forth from the inside to the outside of the sample compartment. And so you can actually image, as the protein moves through solution, what is the concentration at very specific positions in the cell, and we refer to that as the radial position. So that's basically the distance from the inside to the outside of the cell from the center of the rotor. And then at the bottom, of course, the photomultiplier tube is used to amplify the signal, and we get a readout either in absorbance or interference optics to image the concentration gradients or the concentration, the boundaries as they move across the cell. And we'll talk more about each of those concepts. You will see up here, this illustrates the top-down view of a sample cell, where you have two compartments, one of which contains your sample, and one of which contains a buffer blank, labeled as the reference here. And so if you were to assemble the sample cell and look straight through it, if you had something like hemoglobin, you would actually see that the one chamber would be red, and the other chamber containing your standard buffer blank will be clear. And so the light can pass through those windows, and the light will make it through to the imaging setup. Down here we illustrate some of the different components of a sample cell, and then off to the right here you can see that the sample cell assembly is a little bit complicated. In the middle is your centerpiece that contains your actual samples and the buffer blank. That will be sandwiched on top and bottom with windows that are either quartz or sapphire, and then the whole thing is torqued down so that it will be sealed, and you can spin it at up to generally 48,000 RPM, but up to 60,000 potentially. All right, so this is an overview of the sedimentation velocity type of experiment. What you're looking at is a series of sequential scans of the protein taken every 2 or 3 minutes, and you can watch how the protein is actually moving through solution in real time by these different snapshots. And so, for example, this is the first snapshot, and you can see this is the location of the protein at that point in time, and then several scans later it is moved further down, and eventually it will clear all the way through the cell. So here on the left, this represents the portion of the sample compartment that is closest to the center of the rotor, and we're always going to be looking at this from the perspective of the inside on the left, and the protein is always going to be traveling toward the right. And then eventually what happens is the protein will go all the way, will sediment all the way through the sample cell and will crash into the back wall of the sample compartment, and basically that's what's referred to as clearing the meniscus, where we can actually see the protein has gone all the way through, and the only thing left is basically our buffer blank. So the sedimentation velocity experiment is one of the two types of approaches that we can use in the analytical ultracentrifuge. In general, the characteristics of this analytical approach are that we use a relatively high rotor speed, and we're actually throwing the protein outward through centrifugal force, and we're measuring the rate of sedimentation of our protein or other macromolecule. And this yields information on primarily the sedimentation coefficient, but also some very important information about the frictional coefficient, I'll discuss that more later, we actually determine that as a frictional ratio, which I'll explain in a few slides. And from this, we can get an estimate of the molecular weight, and we'll talk more about the ins and outs of how that all works. The illustration I always like to give in my classes is that it's basically like going up to a bathtub or a swimming pool, and if you place your fist in the swimming pool and you move it through, it's going to go through relatively easily. If I then repeat the process and hold my hand extended or rigid, and I apply the same force for my elbow and move my hand through, obviously it's going to go through more slowly because of drag. And it's basically no different for a protein or another macromolecule when you're spinning it in a centrifuge. If you have a protein that is essentially spherical, or like my fist, it's going to go through relatively easily. And if you have a protein that is much more elongated or non-globular, then you're going to see significant drag, and that affects its sedimentation behavior. And that's really a snapshot of what we're doing in a sedimentation velocity experiment. Finally, down here, I'll show you that in the velocity case, we are choosing conditions where sedimentation is the dominant process, and diffusion against that gradient is minimal. And we'll talk more about a different experiment, sedimentation equilibrium, where the balance of those forces is different. You might notice that in a lot of these illustrations, the sample cells are not rectangular in terms of the sample compartments. They have what we refer to as this sector shape. And so you might be wondering why that might be. Well, the reason is that the sedimentation velocity experiment is essentially a kinetic experiment. We're looking at the rate of transport of our macromolecule through solution. So if you look here on the lower right, in this region down here, we can see that if you illustrate it in this way, it makes it pretty clear that when the protein is being accelerated by the centrifugal field, that exists as centrifugal lines of force that emanate from the center of the rotor outward. And so this middle panel shows you that you want to have a sample compartment that is sector shaped so that you can align that with the centrifugal field, and all of the protein will just go straight outward from the center of the rotor, and you won't have a situation such as illustrated in this top portion, where a rectangular containment chamber will actually cause eddy currents to arise, because the protein is going outward and it's crashing into the walls of this rectangular compartment, and that causes the protein to curl inward and get these eddy currents that will mess up our analysis. So that's why we have this sector-shaped cell in the velocity experiment. All right, there will be a handful of you who love equations, probably many more of you who do not particularly care for equations, so I've tried to go pretty light in terms of all the math. But I want to show a few of these important and famous equations to illustrate some of the important underlying principles. I'm skipping over the Lamb equation, which is perhaps the most important one, because that actually describes the process of sedimentation, and I'll allude to that briefly later on. But these are some very important ones. The Svedberg equation shows us that the primary term that we're looking at, the sedimentation coefficient, is proportional to the mass. So that's extremely helpful. That means that the larger the protein or macromolecule that we are analyzing, the larger the sedimentation coefficient will be in general. However, you'll notice that that is modified by the presence of this term, the frictional coefficient. And this is inversely proportional to the sedimentation coefficient. Well, the frictional coefficient F is highly dependent on mass. That is the term that is involved with what I was describing earlier, that my fist goes through solution much more readily than my spread out palm. Or, in other words, a non-globular or elongated protein will have more drag than a spherical protein. And so that just tells us that although, in general, larger mass proteins will have larger sedimentation coefficients, that is not a monotonic arrangement, and if you have things that are different shapes, then that will not have a perfect scaling relationship between S and M, and so it becomes a little more complicated. And so down here in Stokes' law, this shows the relationship of the frictional coefficient to the diffusion coefficient, which is something that we can measure from other experiments such as dynamic light scattering, and we see that the frictional coefficient is related to the overall dimensions, the Stokes radius of our macromolecule. So the bottom line is that keep in mind that the sedimentation coefficient is inversely proportional to the frictional coefficient, but it is directly proportional up here to the mass. And so this becomes an important set of concepts that we will be continuing to talk about today. All right. In terms of a sedimentation velocity experiment, I showed you some sample data earlier. I showed you that we're watching in real time as our protein moves through our sample chamber. What I'm going to show you is a little more detailed way to think about this. On the left is a theoretical experiment. So if we had a global switch and we could turn off diffusion for this thought experiment, this is what you would see. At the very beginning of our velocity experiment, we will have a solid concentration constant across our entire sample chamber. So we load our protein at a fixed concentration, and if we're measuring the absorbance, for example, on the y-axis here, we have a uniform concentration all the way through our sample compartment. Now, as we start to spin the rotor, then we're applying a centrifugal force and we're throwing the protein outward, and then after a certain amount of time, you will see something like this. Again, remember, diffusion has been turned off in our thought experiment here. What happens is that the protein will sediment. Remember that we're always by default showing the inside of the cell on the left and the outside of the cell to the right, so the protein is going in this direction in our schematic here, left to right. We see that at time 1, the protein has moved has moved toward the outside of the cell by this amount, and so what happens is that there is a boundary between the region that contains protein on the right and pure buffer on the left, where the protein has basically left the region. Now we'll note at the top here that we have this plateau that is the general concentration now. You notice that it has gone down. It is lowered. That has to do with the fact that we are in a sector-shaped cell, and so as the protein is going outward, it's going into a wider and wider region of the sector, and that makes it look like a dilution effect here. And then finally, we'll notice that the protein has to go somewhere. We're in a sealed compartment, and so the protein backs up and basically crashes into the back wall of the cell, and that is where you see this spike. And then finally, if we wait another period of time, then we'll see the right-hand situation where we have a boundary, we have a plateau, again, it has diminished because of the wider portion of the sector, and then again, it's moving to the left here because we're getting more and more protein basically slammed up against the back of the sample compartment and just getting stuck there. Okay? So again, this is our schematic, but in real life, it's not true that there is a global switch to turn off diffusion, and so what we really see is something like what I'm showing on the right. We see basically these sigmoidal curves where things kind of get, because of diffusion, we don't see these extremely sharp boundaries. And we'll talk more about the slope of these boundaries in a few slides. That's a very important aspect. And let me just forward it here, and this kind of just backs up what I just showed you. If we take this illustration of a sector-shaped sample cell for velocity, you can see that after a certain period of time, although the entire sample is shown by these delineations, after a certain period of time, you'll notice that the protein is only present in the right-hand portion, and the rest of this to the left here is actually, this is just buffer, and over here, this is all the protein. And what that looks like in terms of the data that comes out is shown up top. It's flat here because that's where we have cleared the meniscus. That's where we have cleared out protein, and it is no longer, there's no longer protein in that portion of the cell, it's just buffer. And then we have this boundary that represents the transition between cleared out buffer and our protein. And then we have the plateau where we have a stable concentration of the protein. Okay? All right. We can think about how would one analyze these type of data sets. And what I'm showing you here is a simple way to think about it, that if you just look at the different boundaries at different snapshots, different periods of time throughout the course of the experiment, well, what happens is the protein is moving through our sample chamber, and we can actually very accurately measure distances in terms of millimeters of travel where that boundary has occurred. So if we look at the upper left, you'll see that there are, this is a very nice illustration from this paper by Berkowitz, but you can see that there are a number of well-defined boundaries that are traveling through our sample cell. We can simply mark the midpoint of each one of these sigmoidal transitions, these boundaries, and then basically look at how far does it move in a given period of time. And you very simply figure out that's a velocity. You can figure out how fast the protein is actually moving through our sample chamber. And then on the right you can see that you can plot on the y-axis the natural log of the position, the location of these green dots. Select my, okay. So these green dots, you can plot the natural log of the position of these green dots. That's the location of the boundary as a function of time times angular velocity squared. So in other words, it is dependent on time, but it's also dependent on the rotor speed. If we spin at a higher rate of rotation, then we're going to throw things out more rapidly. All right. Now, this works quite well if you have a perfectly monodisperse single species in your sample. Then this sort of analysis will work. However, as I'm sure many of you will realize, in real life, it's often not that simple. On the left here is an example of some data from my lab of one of our biofilm proteins, and in this case there's a single species. And so this looks a lot like what I showed you in the last slide. We have these nice sigmoidal transition curves, and if you really wanted to, you could mark the midpoint of these various curves, and then you could look at that the same way as a function of time and figure out the rate of sedimentation. But look here on the right. This is an example from Chad Brodigam's paper down with the reference shown at the bottom. By the way, I will always show the references that are relevant to the data or the concepts on the bottom here. Hopefully everyone has been able to download the PDF, so if you want to dig a little deeper and look into more of these details, you can do that. So Chad and colleagues were looking at a system where there are two different components, and they're looking at their interaction. Well, look at these boundaries. You'll notice that you have a combination of boundaries up on the top here that actually move quite rapidly. And then in addition, you have a portion of your sample that is comprised of boundaries that are very, very closely spaced, and this tells us that whatever's down here, this is a much, much smaller component that is moving very slowly. And so you can kind of have this place this dividing line and see that we have at least two things that are of very different sizes and or very different shapes in our mixture here. And so clearly just marking the midpoint of each one of these transitions is not going to do us much good. So there have been a number of very smart people who've come up with different ways to analyze sedimentation velocity data. And I am not going to be able to go over each one of these because of time constraints. I will introduce some of these. Turns out there are a couple of webinars in particular also sponsored by Beckman that will be very useful for this. And there's a lot of good literature out about these different approaches. I will warn you that, as is not uncommon with scientists, every single method tends to have its group of proponents who are very strongly in favor of their particular flavor. I tried to sort of take a more broad approach here and introduce all of them relatively equally. But I tend to focus in my own lab on one particular type of analysis, which I will focus on today. So very briefly, I mentioned this Lamb equation. This is sort of the master equation that describes sedimentation in a centrifuge. And a lot of these techniques are obviously ways to analyze the Lamb equation. It's complicated enough that we can't really get a detailed analytical solution. We have to assess boundary conditions and have various assumptions or simplifications to allow it to be approachable from a computational perspective. But there are a number of programs that have done very nice jobs of using the Lamb equation and fitting the data to that. So one of the earlier ones that was really powerful is the program SPEDBERG by John Philo. So this is very good for low molecular weight samples. It's also very nice because you can get very good error bars on your sedimentation coefficients. And so if you're doing some of the examples I'll talk about later on, where you're plotting a weight averaged value of sedimentation coefficient to monitor a process such as assembly or binding, then you can actually get very nice error bars on your S values for further fitting. The second completely different approach is the Van Holt-Weishett approach. That's very powerful. One of the earlier methods for looking at multi-component mixtures and distinguishing them, etc. This is actually implemented in the program Ultrascan by Boris Demeler, who has given one of these Beckman-sponsored webinars on lab roots. So I would encourage any of you to look into his webinar for more information on Ultrascan. Then there's a different class of ways to analyze data, generally called the time derivative approaches, G of S or dCdt, so in other words, the change in concentration with a change in time. These are very nice for looking at distributions. What I mean by that, I'll show you in a second, is that if you have a solution and you don't know what is in it, and you want to figure out, do I have a monodisperse solution with a single species, or do I have multiple species? These kinds of time derivative based methods are very nice because they will give you an output that shows you how many different components are in your sample. And then finally, what I'll be focusing on today is this C of S analysis. This is basically something that builds on the time derivative approach, and this is implemented in the program SEDFIT and the companion program SEDFAT, developed by Peter Shook at the NIH. And so we'll be talking a lot about this type of analysis, and most of the data I'm showing you will come from SEDFIT. All right, so I mentioned that we have many times samples that have multiple components. We're trying to figure out how do we deconvolute this complicated set of data with potentially multiple boundaries, all sedimenting at different rates, and understand what what is present in our samples. So the program SEDFIT, as I mentioned, is the one that we often like to use. It's freely available. I believe all these programs that I mentioned are freely available for download, and that's a very nice thing about the AUC community. It's very good for that sort of thing. So this shows you a screenshot of SEDFIT, where at the top you have a series of data curves. Those are fitted, and we see the residuals here. And then what comes out of this is a sedimentation coefficient distribution plot. And I'll show you another view. Let's look up here on the upper right, and you can see some examples from my lab blown up a little bit. What this will tell you is, again, a sedimentation coefficient distribution. So the primary parameter we're looking at here is the sedimentation coefficient. That is related to how fast something will sediment through solution. It is measured in terms of Svedberg units, so many S. So for example, in this plot here, this shows you 3 different samples of IgA, the antibody that was purified from serum from myeloma patients. And what you'll notice is that here we have a peak around 6 Svedbergs, and that turns out to be the IgA monomer. And we have other peaks here that correspond to the IgA dimer. And then in one of these samples, we actually have significant populations of higher order, probably trimeric or tetrameric forms of IgA as well. And then way over here, we see the really massive stuff, and this is generally aggregate or some kind of potentially a circulating immune complex, something really massive that is so large that the program basically says, yeah, it's even bigger than what we're currently looking at. But let me also point out that I'm mentioning that these peaks are 6S or 9S or 12S. The S here is the same one that we refer to when we're talking about the ribosome. The 30S particle, the 50S particle, the 70S ribosome, that S is a sedimentation coefficient in Svedbergs. So it's the exact same approach. It's just that we're talking about individual proteins, so our S values are generally quite a bit lower. So the real advantage, I would say, of using the C of S approach is that you can go in essentially blind. You have a sample, someone hands to you and says, oh, this is really good pure protein. You don't have to take their word for it. You put it in the analytical ultracentrifuge, you do a C of S distribution, and if you see something like this top data set here, you can say, well, you know, your protein is not monodisperse. It may be highly purified, but you have some other things going on here that you need to think about, especially if you're trying to pull out some details about binding constants or thermodynamics. You need to really understand what you're dealing with. All right, I will keep moving on. So let me mention a couple other concepts about sedimentation velocity, and then we'll move on to the equilibrium case. In our thought experiment, shown here on the left, we turned off diffusion. On the right-hand side, we allowed diffusion to occur. But what happens if you have two samples, and let's say they sediment at about the same rate, but one of them is a sphere and one of them is highly elongated or non-globular? Well, as I mentioned earlier, the component that is non-globular will have much higher friction. And so what happens is, because of that additional drag, that additional friction, it turns out that the sedimentation boundary, or the profile we observe, is going to look a lot more like this sort of thought experiment than it will to this sigmoidal curve here. In other words, the slope of the boundary is related to the overall shape of our sedimenting species. And I'm showing you here with these red lines on the right what would happen if we had increasingly elongated sedimenting species. We will see a steeper and steeper slope of the boundary. Okay? And so from that, we can actually determine the so-called frictional ratio. This is the frictional coefficient of our sedimenting species compared to that of an ideal sphere of identical volume. Now, many of you will probably be already starting to formulate this question when I say the shape of your species. If you have multiple components, then that can be a challenge. This approach works ideally when you have a single species, and you can get a single boundary, and you can get a slope of that boundary, and we'll talk more about cases where that's not the situation later on. Let me give you some examples from my own lab. On the left is the FC fragment of IgA1, and you can see, if I click on here, I hope that shows up, that it has a slope as illustrated in the gray dashed line, and I've highlighted with red. On the right-hand side, we have another protein. This is one of our biofilm proteins that is basically this elongated thread-like protein. It is very, very, very elongated, and so this is clearly a very non-globular situation. If we look at the slope now of the boundaries with this highly elongated protein, and then I'm going to, for comparison, illustrate the gray slope here. That is exactly what I showed you on the left-hand side. You can see that there's a very distinct difference in the slope of these boundaries from our more globular protein, FC alpha, and our less globular or highly elongated protein over here, the biofilm protein from the protein AAP. So the reason I'm going through all this in a little bit more detail is that this is what illustrates the concept I made at the very beginning with the Svedberg equation, that the sedimentation coefficient is proportional to mass, but it is influenced by this frictional ratio. But if we know S, which we will, and we can determine the frictional coefficient, then from those two, we can directly calculate this buoyant mass, m sub b, which is the actual mass times some correction terms, the v-bar and rho, which are the partial specific volume of the protein and the buffer density. So those are our parameters that we can get easily. So if we can accurately measure the frictional ratio, then that combined with our S value lets us estimate the mass reasonably well. One thing that is an important corollary is that if you have two samples, whoops, I apologize. I'll do this. If you have two samples that have very different shapes, you actually can potentially have something where they co-sediment. They sediment at the same rate, even though they have widely different shapes and different masses, because of the effects of this frictional coefficient and of drag. And I'll talk at the very end of the seminar about how one can resolve situations like that. All right, so just to wrap up the velocity part of the intro here, velocity looks at snapshots of the protein. It's a kinetic experiment. We're looking over time as the protein moves through solution. It gives us information on size and on shape, And if everything aligns properly, we can get some pretty good idea of the mass. And from deconvoluting the data on the left, we convert that to a sedimentation coefficient distribution. And this is showing 3 different samples superimposed for comparison, but you get these essentially Gaussian curves that will show you what sedimentation coefficients are present for the components of your sample. All right, let's move on to a highly complementary technique that is also done in the analytical ultracentrifuge, and that is called sedimentation equilibrium. So in this experiment, we tend to do several 3 or 4 lower rotor speeds, and we do much longer runs. The velocity experiment is generally overnight, so 14 to 16 hours much of the time. With the equilibrium case, we will usually start at 14 and go up to 24 hours. This really depends on the viscosity of the sample and potentially on the shape of the sample, how long you must sediment. But what happens is we have obviously centrifugal force acting on our protein, and so sedimentation is throwing our protein outward toward the outside of the cell. And any time in chemistry that you build up a concentration gradient, we're throwing protein to the outside of the cell, that's building up protein in the back here, we have a concentration gradient. So then you have diffusion against that gradient, okay? So diffusion will oppose sedimentation. And if we let this spin and spin and spin for, let's say, 24 hours, eventually what happens is the force due to sedimentation and the force due to diffusion will equilibrate, and we have no net movement of our macromolecules, and everything settles down to a stable equilibrium concentration gradient. And that's what's illustrated up on the left here with at 4 different speeds. You can see that we have these exponential curves that are our stable equilibrium concentration gradients. And as I'll show you in a moment, by fitting those exponential curves, we can directly pull out the molecular weight of our sample without any effects of shape and without any standards, etc. It's an amazing first principles, highly applicable technique. So just to reiterate, if we start the experiment, it's going to look a lot like our typical sedimentation velocity types of experiments, okay? So we'll see that we have this boundary that moves through the cell over time, we have a plateau, and then the sample crashes into the back of the cell. But what happens is if you keep this going and keep this going and keep this going, then eventually, as I mentioned, you'll get this stable concentration gradient that will look more like this. It looks like an exponential curve. So let me explain a little bit more about a few more equations. I'm sorry. This is a really very nice analysis by Tom Lowy and Walt Stafford in this review. It came out a number of years ago, but this is a really fantastic review. The reference is down here. I'm working on my whiteboard skills. Sorry, guys. Okay. So it's down here. I've adapted some of what they had in their paper to make it a little better applied to this particular webinar format. But this really comes from Tom and Walt. So if we look up here in the center, we see an illustration of our sedimentation equilibrium experiment. You can see the exponential curve that is our stable concentration gradient. And in the middle, you'll see this plane, which I'm highlighting with my red lines here. So you can imagine that there's a plane through the middle of our sample. And the red arrow shows you the flux due to macromolecules that are being sedimented. So molecules will cross through that plane, and we're going to count how many are crossing through that plane. Okay? And then in blue, the blue arrow here shows you the flux due to diffusion. So these are the macromolecules that are diffusing against the gradient and are crossing that plane. All right, so we have these 2 fluxes due to sedimentation and due to diffusion, and we know what the equations are that describe these 2 fluxes. For sedimentation, the flux is the concentration times the velocity. In the radial acceleration of the rotor, it's expressed as in this way, c times s times omega squared r. Over here we see this is the equation for flux due to diffusion. It's simply the diffusion coefficient d times the gradient, dc dr, the change in concentration with the radial position. Okay, but If we let this go until these two forces have equilibrated and we have a stable concentration gradient, then by definition, if it is truly at equilibrium, then we can say that the flux due to sedimentation and that due to diffusion are and must be equal. So we can equate these two terms as shown here. At the bottom, we're simply rearranging terms. We're putting the R, all the R's on the right-hand side. We see this equation here. We can do some very simple differential calculus and figure out that this is our equation. I'm going to go to the next slide and just reprint the same equation. So what we're going to do now that we rearrange these terms is define this parameter called sigma, which is the reduced buoyant molecular weight. And there are two ways to look at or think about sigma and what it means. On the left-hand side, we can think of this in terms of an experimental definition. This term tells us that sigma describes the curvature of that stable concentration gradient. It's a change in the natural log of the concentration as a function of the radial position. Okay? So it describes the shape of the curve. But what's really cool is that sigma, using the other equated term here, gives us the molecular definition. So sigma is directly proportional to the mass. All these other parameters are things that we know that are experimental. So V bar is based on primarily the protein sequence. That's the partial specific volume. Rho is the buffer density. We can calculate or measure that. Omega squared is related to the rotor speed. And RT, this is the gas constant, and temperature in Kelvin. So all of these other terms we can figure out. Once we know sigma, we get the mass. And this is a very powerful thing because this is not dependent on standards. This is not dependent on shape. If it is truly at equilibrium, you can have two things that are completely different shapes, but if they have the same mass, they will give rise to the exact same equilibrium concentration gradient because it is solely dependent on mass after these additional terms have been accounted for. So to restate what I just told you, this means that the curvature in the equilibrium curves or the equilibrium gradients directly gives you molecular weight. No standards, no matrix, so this is the advantage, one of the big advantages of AUC over something like gel filtration. We're working in my lab on a protein with a biotech company, and this particular protein will stick to the matrix, and it messes up your gel filtration experiment. We can use a wide range of buffers. You can change the salt, you can change the pH, you can change the actual buffer component, and you can change the temperature anywhere from about 4 degrees to 40. So you can actually get a wonderful amount of information from these combinations of techniques. Secondly, if you have a system that is undergoing self-assembly, so monomer to dimer, etc., which we'll talk about later, then you can actually directly pull out the equilibrium constants for those transitions between the different oligomeric states. All right, here's an example also with our biofilm protein where we're showing 4 different speeds. Generally speaking, the way you approach this is you will take 3 concentrations of your sample, generally over about a 10-fold concentration range when you load, and then you sediment them so that they will achieve their stable concentration gradients. So your 10-fold range in concentration that you loaded then basically ends up becoming about a 100-fold range in concentration after the gradients have been achieved. So you're really covering a wide range of concentrations. Then we'll also spin those samples at 3 or 4 different speeds. So we end up with 9 to 12 curves typically that we're trying to fit all at the same time, so global analysis to pull out the information we want to get. Well, why are we going to all that trouble? Well, what I'll show you in the next slide is that the data we observe is really the sum of everything that is sedimenting in our sample. And if, for example, we have monomer, dimer, and tetramer of our protein, then each one of those will give rise to its own stable concentration gradient depending on its own molecular weight. So the monomer has a mass m, the dimer has a mass 2m, The tetramer has a mass 4m, and so each one of those will give rise to a different shape of concentration gradient. But what we observe is the sum of all those in solution at the same time. And so the challenge is that the sum of a series of exponential curves looks an awful lot like an exponential curve. And so it becomes a little bit hard to deconvolute this, and the way we get at that is by doing global analysis and pulling out all the different information. It looks like I need to speed up a little bit here. So this is an example shown from some early work in my lab with, actually my thesis work with FGF on the right-hand side, and you can see that the observed data in diamonds then can be deconvoluted into the components, which in this case were monomer and dimer and tetramer, and so we can actually pull out the concentrations of these components, and then you're changing concentration in your different samples, and you're changing the distribution by changing the rotor speed. So then you're actually watching shifts in the system as at low concentration and low speed, you might have primarily monomer. As you go to higher speeds and higher concentrations, that equilibrium will shift toward dimers and toward tetramers. So that's the power of this technique. You're actually observing a wide range of behavior of your system, and that's why you can actually analyze your data and get accurate equilibrium constants that govern the transition between the different oligomeric states. All right, now we'll get to what I really wanted to talk about, which is how is AUC complementary to structural biology? My other hat is that I'm an X-ray crystallographer. We do a lot of other biophysical techniques, as Chad mentioned in the intro. And so we have found that AUC is an enormously beneficial tool for the structural biologist. And so I'm going to really kind of hit on a number of points. First of all, analyzing situations where you have monodisperse samples, and I'll give you multiple examples where someone, either ourselves or a collaborator, had crystallized something, and this is what you see in the unit cell, but what do we actually see in solution, and how do we characterize that? Secondly, we can use AUC, as I already briefly mentioned, to look at multi-species mixtures, and it's a very powerful way. If you think you have a monodisperse sample, you might not, and this is a great way to find out. And then we'll spend some time talking about some of the more interesting ways to use AUC, such as with interacting systems, whether we're talking about self-assembly or heteroassociation, like different proteins interacting. And then I'll finish with a couple of just caveats, things to keep in mind about Gilbert-Jenkins theory and reaction boundaries, and then finish up with a really cool new approach that's a two-dimensional size and shape analysis. Okay, so that's kind of the remainder of what we're going to talk about. Let's start with AUC on monodisperse samples. So one of the important criteria for success, especially as a crystallographer, but I think with a lot of structural biology techniques, is to have a highly purified, preferably monodisperse sample. And so if you have a mix of protein that is forming monomers and some higher order oligomers, that might be okay. But if you have a protein and it goes to aggregation, then this is a real problem, and you're basically never going to get that to crystallize, or at least to form highly diffracting crystals. Okay, so this is sort of, from a sample quality perspective, an important issue. Number two, this is really well, this is a very common occurrence, that in crystallography you will often see what looks like a multimer in your structure. So, okay, this looks like a dimer. But of course, crystals depend on having proteins rubbing shoulders and bumping elbows, because you have to have the crystal lattice forming by contacts between proteins. So these crystal packing contact points may make it appear that a protein is forming an oligomer, when in fact that's really an artifact of crystal packing. So a lot of times we'll see something that looks very interesting in the crystal. We want to verify that in solution and say, well, it looks like a dimer in the crystal. Is that real in solution? In terms of NMR, assessing the assembly state is really critical, because if you have a protein that's right on the edge at which you can measure, let's say 35 kilodaltons, and the boundaries keep getting pushed further and further, but a few years ago that was kind of about as large as you could probably analyze by NMR for multidimensional structural NMR, 35, 40 kilodaltons. But you then find that your protein's a trimer. Well, now it's 120, and that's going to be nearly impossible to analyze, so that's very important. And even in cryo-EM, a lot of times you'll see situations where you have these oligomeric assemblies, and you really want to understand exactly whether that is a pentamer, a hexamer, a heptamer, octamer, so you can do your averaging and increase the signal to noise. So now I'm going to blast through a whole bunch of examples. These are cases where my lab has used AUC to verify apparent multimers by crystallography. So I'm not going to spend a lot of time on each of these, but the references are shown. On the left, we see convulsin. This is a lectin-like protein from South American rattlesnake venom. But it turns out it's an extremely potent agonist of platelets because it binds to and activates the platelet collagen receptor called GP6. Now, it had been shown by a couple of groups when they saw the structure that this forms this really interesting heterooctameric ring. So alpha, beta, alpha, beta, alpha, beta, alpha, beta. And I'm showing you that in tan. Turns out in the crystal structure, there was a second ring that was back-to-back with the first ring, but they just assumed that was crystal packing. They never mentioned it at all in the paper. Well, when we started to analyze this, because we wanted to look at the binding to GP6, my postdoc Katsunori Hori did the AUC and found out to our surprise that actually that stacked donut is really the normal state of convulsin. There are two heterooctameric rings that stay together and are assembled as this double octamer. So that was really important to understand, or to interpret our binding data and understand why this is such a potent agonist. On the right is work from Chuck Bell at OSU. This is the REC-E toroidal exonuclease, and the structure clearly showed it formed a tetramer. And that was very interesting because a structure by my close colleague Brett Koval here in Cincinnati years before had shown that this other well-known toroidal exonuclease was in fact a trimer. So we wanted to double check and make sure that this wasn't something weird. And so AUC confirmed Chuck's structure that REC-E is in fact a tetramer. And so it makes for a kind of interesting question about why these two different folds evolved in two different quaternary forms to achieve a similar kind of function. So another example would be metal-induced assembly, and this has been an interest in my lab. On the left, we see a calcium-activated nucleotidase. This is a collaboration between my lab and Terry Curley here at Cincinnati. Terry had found that in the presence of calcium, you see some different spectroscopic signals with this nucleotidase, and it changes dramatically, increases the activity. And Katsunori in my lab figured out that it was actually inducing dimerization. And so you can see that by the sedimentation velocity experiment, and then we also followed up with some equilibrium data. On the right is an ongoing, really neat story from my lab of this biofilm related protein called AAP. So Deb Conradi and Kristen Brescia in my lab have been working on this, and Deb saw the structure, and you can see that this actually assembles in the presence of zinc. And after a lot of hard work, she was able to get the structure of this dimeric construct of this region of AAP. But really the important experiment was quite a bit earlier when Kristen and Deb did the sedimentation experiment, and they found that this protein is a monomer in most cases, shown here. But in the presence of zinc, we get this pronounced shift to the dimeric form. And that turns out to be critical for the formation of these staph biofilms. That if you have this, you have zinc allowing this protein to self-assemble at the surface of the bacteria, that allows basically the cells to stick to one another and to form this biofilm that eventually ends up being highly resistant to antibiotics and to immune cell responses. So that was a really neat, really neat experiment. And then we also have several collaborative projects with Pingwei Li at Texas A&M, who works in innate immunity. And so this just shows that we've used AUC to verify, for example, that the RIG-I RNA sensor forms a 2-to-1 complex bound to RNA. On the right-hand side, you can see this is a neat story with cyclic GMP-AMP synthase, or CGAS. The structure actually, once again, showed a 2-to-2 complex. A lot of the previous people working in the system had assumed that was just crystal packing, but we actually verified by AUC that no, that dimer is actually forming, and higher order oligomers are forming in solution, and Pingwei was able to show that's actually absolutely essential for the activity of this important enzyme. All right, let me go back to something I've already alluded to a bit, and this is very powerful for a structural biologist. Again, if you have a purified protein, it's a single band on a gel, it's easy to say, I'm done, it's 98% pure, but if you really have a mixture of monomer, dimer, tetramer, and aggregate, well, that's going to be a problem. And AUC, especially the CFS analysis by sedimentation velocity, is a very powerful way to detect that. Anytime you have a new sample, this is really the first thing we always do is run it on the AUC. Anybody here who's from the biotech or pharma world, this is extremely powerful. And we're doing some work with some biotech companies, and it's been very useful to do quality control on different batches of proteins. And we're looking for things like aggregates, if you have any kind of biological, an antibody or otherwise, because that can really torpedo the therapeutic efficacy and cause some really dangerous side effects if you have aggregate. And so it's a very powerful way to look at that. And even for sort of early stage comparison of different batches, different lots, or as you're approaching formulation studies for early stage development, AUC is a very powerful technique for that sort of approach. All right, in the time I have left, I'm going to talk about some more of the interesting details, and I will necessarily need to go relatively fast through these. But in addition to initial characterization, AUC is a wonderful technique for really delving into the finer details. The quantitative aspects of protein assembly or protein ligand, protein-protein interaction, stoichiometry, and really getting at things like accurate equilibrium constants. So one of the primary ways that AUC is used is to look at equilibrium assembly, in which we have a monomer that is assembling to a dimer or a tetramer or whatever other thing, okay? So from especially the equilibrium experiment, we can determine the equilibrium constants. From both equilibrium and velocity, we can determine stoichiometry of oligomers. And this is similar to what I talked about earlier with the case of convulsin and Rec-E, where we had a stable oligomer, but those were very high affinity oligomers, and so what we were observing was a stable oligomeric state. But in addition, you'll have situations where the concentration regime over which that assembly occurs is right where we're able to access experimentally. And so that's where you're actually able to get information on accurate determination of these equilibrium constants. Real quick, here's an example, also a collaboration, a three-way collaboration between Allison Weiss and Rhett Kobal in my lab here in Cincinnati. Allison's worked for many years on different bacterial toxins, and Shiga toxin has Shiga toxin 1 and 2. If you saw the crystal structure, the B pentamer portion of that toxin always looks like a stable pentamer. But when you do the actual AUC experiment over here, you'll see that Shiga toxin 1 is highly stable, and it's pretty much sedimenting as a mostly pure pentamer. Whereas if we repeat the experiment at similar concentrations with Shiga toxin 2, we see a whole range of species from monomer all the way up to pentamer. So it's clearly a much weaker interaction that stabilizes the pentamer in Shiga toxin 2. And there's probably some really important significance in terms of the biology to that phenomenon. And then we used equilibrium to really figure this out in detail. And so I'm showing you one single equilibrium gradient curve. On the left is Shiga toxin 1b, and if you can see that black dashed line over here, that basically is showing you that the sample is almost entirely pentamer. On the right-hand side, you can see that this light gray curve is monomer. So most of our sample in this condition is monomeric, and just a small amount down here is the pentamer. And so from these kinds of experiments, we pulled out the equilibrium constants for both Shiga toxin 1 and Shiga toxin 2. One quick point I'll make that's a nice feature of the analytical ultracentrifuge is that if you choose an appropriate buffer, such as a phosphate buffer, it's highly transparent. And so we've been able, in a couple cases, to go down to very, very low wavelengths, as low as 203 nanometer, to get very, very high sensitivity and really look at extremely low concentrations down to the low nanomolar range. But you have to have a really clean buffer and not have a lot of extras or reducing agent or EDTA and that sort of thing. All right. This is one of my favorite topics, but I'll have to go quickly over it. This is a really neat way to analyze data. In this situation, you have what's called a linked equilibrium. So we have two things going on. In our case, we have proteins that bind to a metal, such as zinc. Now, that's not something we can generally follow by AUC because the addition of a single metal to a protein is not going to change its mass or shape, usually, very dramatically. But when you have something where the zinc binds and it induces dimerization, that's a linked equilibrium because we have M binding to X, in this case zinc, that is sort of an invisible equilibrium, but that is linked to this highly visible equilibrium of the dimerization. So we can repeat the dimerization experiment at different fixed concentrations of zinc, and each one will cause a different shift of monomer toward dimer, and eventually, if we have enough zinc, we'll go all the way to forming the dimer. Well, the math here, in terms of the thermodynamics, is really neat, because what you can do is you can take data like this. You'll see these sigmoidal transitions where the protein goes from monomer up toward dimer as a function of zinc. You can fit this to get the equilibrium constant from sedimentation equilibrium experiments, and then take this plot here where you plot the log of your equilibrium constant, so the dimerization constant, as a function of different zinc concentrations. Plot those log K values versus log of the zinc concentration, and you'll get a slope. And the slope of this linear response tells you the net number of zinc ions that were bound or released upon dimerization. So it's an amazing way to get some really detailed information about your system from a few well-conducted, carefully conducted AUC experiments. And I always tell my students that this was something we figured out years before we finally were able to crack the crystal structure. And it basically showed us years ahead of time what we eventually would find from doing the structure. This is another example in terms of pH. If you take the dimer form of our protein in the presence of zinc and you lower the pH, then it falls apart and you see it shift. And you can also plot this to get the pKa and do the double log plot to get the net number of ionizable residues that are involved in this dimerization event as a function of pH. All right, in terms of interacting systems, there are a lot of ways to go about this. I've talked a bit about the equilibrium assembly case. What about heteroassociation? So we're not talking about monomer dimer, we're talking about protein binding to another protein, A binding to B, for example. Okay, there are a lot of ways to do this. It's a little more complicated. Probably the simplest is to repeat your experiment, titrate in your ligand B at different concentrations, and plot the weight averaged sedimentation coefficient as a function of concentration, just like a typical binding curve. Okay. You can also do more sophisticated things where you take your data from SedFit, import it into SedFat, and you can actually directly analyze the boundaries and fit those to equations to look at, to get out affinity and stoichiometry information. Sedinal is a very nice program that uses time difference velocity traces to, and it's very powerful because it can look at a lot of different models for heteroassociation. And then there are a lot of other options, but actually Jim Cole has a really nice paper in Methods of Enzymology here that I'm showing at the bottom. For those interested in these techniques to look at heteroassociation, I highly recommend Jim's paper because he talks about some of the more detailed ways to do this. So real quick, here's some other examples from our lab. With heteroassociation, this is an example where we have a receptor and we have an intact antibody. And when you mix the two together, it will shift to the right and you'll see complex forming. So at the very basic level, that tells us something has happened. And then at a more sophisticated level, you can actually start to look at the predicted masses if it is appropriate. And that can help us get at the stoichiometry. In this case, it helped us to show that two Fc alpha receptor 1 molecules bound to a single IgA Fc fragment. Another example is with our GP6 receptor. We titrated in the collagen peptide, and you can see that the apparent sedimentation coefficient would shift to the right as we titrated in the peptide. We got lucky in this case because the peptide does not absorb at 280, and so all we're seeing is the protein as it moves to a larger size, and then you can fit this, like I mentioned, fit the weight average sedimentation coefficient to a binding curve and get the KD of that interaction. In the last several slides here, there are some really new, relatively new and really powerful ways to use the different absorbance and interference capabilities of the Beckman XLI instrument to get at complicated stoichiometry. So you can do multi-signal analysis where you're actually looking at two different components that either absorb differentially, or one of them you follow by absorption, one you follow by interference. And you can actually use SedFit to keep track of these distinct species based on their unique spectroscopic signals. And then you can see here that this shows us there's a peak, but you can look at the components of that peak, how much of A and how much of B are actually present in this complex right here. And that's a really neat approach I think is going to have increasing use. A couple of caveats, and then we'll finish up with this new 2D size and shape approach. One thing that I think puzzles a lot of people who are new to sedimentation velocity is something like what we see in the upper right. This shows our biofilm protein, the presence of zinc. It's a dimer. And this shows over here the presence, the monomeric form in the absence of zinc. If we go to an intermediate zinc concentration, a lot of people would expect what happens is you have, let's say, a dimer here and your monomer here. And then if that falls apart, then the dimer goes down and the monomer goes up. Well, that can happen if you have very, very slow reaction kinetics. But usually what happens is instead, you'll see something like we see in the upper right, where the peak of the dimer will shift down to the monomer or vice versa. And so we're not really seeing our protein fall apart into a 1.5 mer. That makes no sense. Really, this is due to Gilbert-Jenkins theory, and the fast reaction kinetics will give rise to this apparent shift between the two extremes, because what we're really observing is not two distinct species, but a reaction boundary. So what we mean is that if monomers and dimers each sediment at different rates, which we know is true, if they're frozen or have very, very slow kinetics, then the two boundaries are essentially independent. But if they are in rapid exchange, then as they are sedimenting, monomers are shifting to dimers, dimers are shifting back to monomers, and so it all kind of bleeds together, and that's what we call this reaction boundary. So here's a word of caution. In the upper right, I'm showing you the data again for this shigatoxin 2 B pentamer. The equilibrium data indicates that we have monomer up here, and we have pentamer. We don't see any reason to fit additional species. We can fit the data perfectly well with a strict monomer pentamer equilibrium. But if we look at the bottom here by velocity, it looks like we have all these intermediate peaks that someone who's new to the game might say, oh, we have dimers and we have trimers and we have tetramers. Well, probably not. It's probably just monomer going to pentamer, but it's in fast exchange, and so it looks like you have these intermediate species. Keep that in mind, because that will come into play in a few slides as this caveat. The last part I want to talk about is a relatively new approach that I think is extremely powerful. It's going to get a lot more use, I believe. It's the so-called 2D size and shape distribution. So this is implemented in SedFit, and as I'll mention in the next slide, in a couple other programs as well. So we did a test experiment in my lab. We took this extremely elongated protein from the biofilm, the biofilm protein, and we took lysozyme, which is more or less globular. And we put about 10% of the elongated protein, 90% of our globular protein, and we sedimented it. And it turns out when you do the standard analysis, it looks like we have one species. And we did this on purpose because this is one of those examples where the two proteins are completely different size, completely different shape, completely different mass, but they actually sediment almost at the same rate because of the differential effects of friction. And so how do we resolve these kinds of complicated systems? Well, this new approach by Peter Schuck implemented in SedFit lets you basically analyze your data on a grid where you look at discrete values of the sedimentation coefficient and the frictional ratio at each point on the grid, and it basically allows you to separate out species that are sedimenting very close to one another but have very distinct shapes. And so you can see here in this plot by my student Catherine that we can clearly distinguish the primary species on the left, which is our globular lysozyme, and this little blip over here, which is our highly elongated protein. I definitely want to point out that in Boris Demoler's webinar sponsored by Beckman, he discusses a similar implementation, not the same thing, but similar in Ultrascan, and there's a paper by Carney et al., those are shown over here, that describes a similar approach. But let me show you one example from my lab where we had a really complicated mixture. We had a whole lot of things going on. And if we do the standard CFS analysis, it's shown in this sort of green curve right here. This is the C-gas enzyme plus DNA. There were a lot of things happening. And in order to really figure out what was going on, we used this two-dimensional size and shape approach. And what's really neat is that this enabled us to see that we had a mixture of, let me switch here, things like the DNA, based on the size, we could figure out that was there. And we had a series of compact globular species. Then they started forming these intermediate species that are actually much more elongated. And then we had a few low-level species that were extremely elongated, and possibly even fiber-like type of species. And so this allows us to make some suggestions about what it appeared to be in terms of the stoichiometry. This is, again, that caveat from the Gilbert-Jenkins slide, that remember that if this is an assembling system, we are actually very likely looking at reaction boundaries. And so we have to use caution with these as kinds of assignments, because what looks here like a 2-to-2 or 3-to-3 complex may actually be just a reaction boundary on the way to something to the terminal complex. However, I think anybody who has a really complicated mixture of a lot of different things, this is a fantastic way, and really I think the best way there is to resolve things of very different sizes and shapes. All right, I'm going to wrap up here. And actually, one thing I want to mention, I realized late last night that I neglected to add one more point, so I want to throw out a name. One other powerful way to use AUC for structural biology is to actually use it as a direct method to get hydrodynamic information that can be combined with other techniques such as small angle neutron scattering and small angle X-ray scattering to inform modeling approaches to build up structural information on complicated systems. So I think one of the guys who's done some beautiful work in this area is Stephen Perkins at University College London, and I'm sorry I meant to include a slide on his work. And also there are programs such as HydroPro by Garcia de la Torre that will allow you to do bead modeling based on your sedimentation velocity data. So I definitely want to put a plug in for those kinds of approaches as well. That's where AUC is directly used for structural biology types of investigations. All right, I'm sorry I went a bit over, but to conclude, AUC is an amazing technique for analyzing molecules in solution. It is highly complementary to structural biology in multiple ways. It's also, I think, one of the best techniques for biotech and pharma. And so hopefully this will have spurred some ideas from many of you, and you can find your local AUC person either at your company or your institution or across the town, and you can really do some neat experiments. I just want to acknowledge a lot of wonderful work from people in my lab over the years. The ones I listed here are people whose data I showed. Definitely want to thank Beckman for sponsoring this webinar and for a lot of the great illustrations that they provided. And then a lot of just wonderful sort of giants in the field that have given me advice and help either at workshops or at the Gibbs Conference on Biological Thermodynamics that I go to each year, and Irby Purdue, who set up our early instrument and has given a lot of advice. So thank you. I'd be happy to take questions, and I will wind down here.
Great, thank you for that highly informative presentation, Dr. Herr. Before we get started on the question and answer session, I would like to remind our audience that you can submit questions by typing them in the Q&A box at the lower left of the presentation window. We've already had a lot of great questions come in, so let's get to it. Our first question from John Anders at Anders Therapeutics is whether or not you can assess the percent relative abundance in each peat within a sample.
Yeah, that's a great question. It's a very important point. Yes, you can. I think that's one of the perhaps most powerful advantages to analytical ultracentrifugation, especially when you're looking at complicated samples. So, for example, if you have a protein that you are, let's say, working toward preclinical trials or even hoping to work on formulation, heading toward FDA approval, etc., you really need to understand with accuracy the relative, the proportion of, let's say, monomer or some other oligomers, and especially aggregate. And in comparison to techniques such as dynamic light scattering, at least the raw data from DLS, that's an issue because the signal is dependent on the size of your the component in solution. There are ways you can correct for that, but it basically is modifying your raw data. Whereas with AUC, the signal is based on absorbance or interference optics, and so it's really the extinction coefficient of your protein that defines the signal. And so if you have one protein that is forming aggregate and forming monomers and forming dimers, Absolutely. You can actually look at that and determine the relative proportion of each peak and figure out its relative contribution to the total in your sample. Technical. Great. Sorry about that. I think we had a slight technical issue with one of the PCs. There was a question. I will relay it in case you all did not hear Chad. Someone asked whether the achieving nanomolar concentration is possible with absorbance only in AUC. Oh, in absorbance only AUC. Well, there are ways to do fluorescence analysis. That is a specialized thing. I know that there are some aftermarket things. I actually need to talk with my Beckman colleagues to know what's current. So there are some specialized ways to use fluorescence, but in general, absorbance is probably the best way to go. As I said, I would guess that most of the time people are analyzing proteins in the micromolar range. Interference optics is nice because it lets you go actually higher beyond what would be the linear range of the absorbance spectrophotometer. And so interference allows you to go higher for sure. It really depends on the absorbance of your protein or the interference, the DNDC of the protein, which is what we use in interference optics. But for very low concentrations, if you have a clean buffer, so we're talking phosphate, we're talking no reducing agents, other buffers such as tris generally are not going to work all that well. EDTA is probably going to be a problem, but if you can get by with something like a clean PBS type of buffer, then you can actually go quite low with absorbance. So I hope that addresses that. I'm going to go ahead and see if there's any other questions that jump out at me, and then we'll see if we can wrap up in just a moment. Give me 2 seconds. Someone asked, how do you know or how can you determine the best speed for the protein to be analyzed by AUC? There are two answers to that question depending on the technique you're using. If you're using velocity, we generally, most of the time, will go with 48,000 RPM, which is what our centerpieces are rated to. There are some fancier centerpieces that can go to higher rotor speeds, but the standard ones go to 48,000. And most of the time, that's going to work for almost all proteins. Sometimes if you have something extremely large, that will cause the boundaries to be very well separated, and it kind of screams through your sample compartment and sediments all the way through a little more rapidly than you would like. And in that case, you can cut back the rotor speed to maybe 36,000 or so. But it's pretty forgiving. Now, the second part of this is with sedimentation equilibrium, it's actually very important. And there are some rules of thumb. So actually, anybody who wants to know about this, you can email me and I'll give you my website. I have a little, an old PowerPoint presentation on there. But I'm pretty sure that the Lowy and Stafford article that I mentioned in annual reviews goes over this. But remember that the value of sigma, the curvature, is dependent on the rotor speed. So there are some pretty important rules of thumb to get an ideal range of shapes of the curve. The bottom line is you don't want to have a lot of really shallow curves for an equilibrium analysis. You want to have some that are relatively shallow, some with more curvature, and some that are quite curved, and do the global analysis of all of those. And so let me see if there's any other things that jump out here. I think I'm only seeing some of these questions. All right, let me just finish up with one more. There is a question, is absorbance enough or is interference also needed? You know, this really depends on your particular needs. We honestly do a lot of absorbance, even though we have the option for interference. I think as we do more and more interference, we'll probably see its benefits in some ways. A lot of people who do it continually point out that interference is very good because it has higher precision. It's nice because something that does not have absorbance will show up through interference. And it's nice because you can go to very high concentrations. So those are some real benefits. The challenges are that everything that is in your solution will absorb. And, will give a signal through interference optics. And so if you have a buffer mismatch, that can cause all kinds of conniptions when you're using interference optics, whereas absorbance is much more forgiving. So if we're getting a lot of samples from outside people and we're not sure how close the buffer matches the sample, absorbance is usually how we will go. Or if we're looking at things where you have a protein that absorbs, and like our collagen peptide that does not, it sometimes can be easier to use absorbance at a selective wavelength. So it just really depends on the situation. I think that you can go very far with absorbance only. If you can get interference, it's fantastic because it gives you some other capabilities as well. And I will stop answering questions there. If you have further questions, I will try to answer things that have been posted later on through email. Otherwise, my email address is here on the screen. Thanks.
Okay, thank you, Dr. Herr. That was a great presentation. I also want to thank Beckman for making today's educational webcast possible. Today's webcast will be available for on-demand viewing for 6 months. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thanks a lot and see you next time. Goodbye.